The Purdue Global Science Center presents Antibiotic Resistance. Antibiotic resistance became a priority area of research all around the world over the last few decades. In order to understand how resistance occurs, we will first discuss how antibiotics work. Antibiotics are a class of antimicrobial agents administered to combat infectious living organisms. This includes fungi, protists, and bacteria. Though the focus of this presentation will be with regards to bacteria, it must be noted that antibiotic resistance can occur in all protists and fungi. Bacteria are one-celled organisms that live naturally in all environments, including on and within the human body. Many of these bacteria are harmless, but some are harmful and cause debilitating and life-threatening infections. Antibiotics can be administered topically, orally, through injection, or intravenously. There are many different forms of antibiotics, all aim at eliminating the microbial infection. The first mass-produced antibiotic was originally discovered in 1928. Alexander Fleming is credited with discovering the first antibiotic, penicillin. He was inspired to seek better cures and treatments for bacterial infections due to his battlefield experiences during World War I. He tested a variety of substances by growing bacterial cultures in petri dishes, and then exposing the colonies to different substances, even including tears, which contain a natural enzyme that prohibits bacterial growth. While he had limited success, in 1928 he fortuitously discovered that a species of mold, which was a contaminant within his experiment, prevented the growth of staph bacteria. The petri dish here shows bacterial growth in the form of a cloudy haze. The white discs contain substances that may prevent bacterial growth. Notice that most of these display a halo clear of bacteria. Fleming observed a similar pattern in the dish in which he found the fungus growing. He further experimented with this mold called Penicillium notatum and published his results, but was unable to successfully mass-produce and adequately test penicillin. His work was further advanced by the biochemist Ernst Chain, who was ultimately able to mass-produce and test the antibiotic by injecting it into mice. Penicillin became available to humans worldwide by the end of World War II. Since the discovery of this first widely available antibiotic, numerous others have been discovered, and their uses go beyond human applications to veterinary medicine and in the raising of livestock. Though various antibiotics have been discovered since the 1920s, they all function in similar ways to eliminate infections. Some antibiotics target specific strains of bacteria, while others target general classes or groups of bacteria. They may compromise the cellular structure of the bacteria and therefore destroy them, or they may interfere with the life processes of the bacteria. They may inhibit reproduction, disrupt energy pathways, or prevent the production of proteins by the bacterial cells. All of these approaches ultimately prevent the bacteria from reproducing or surviving and therefore eventually eliminate the infection. Antibiotic resistance can develop in a variety of ways. The natural process of genetic mutation can cause the development of drug resistance. Through reproduction, which requires the replication of DNA, mutations or changes in the DNA can occur spontaneously, and these mutations have the ability to make bacterial cells resistant to a particular antibiotic. These bacteria are therefore not destroyed, and they can lead to a resistant infection. Bacteria also have the ability to transfer segments of DNA amongst one another. This can result in the spread of a gene that causes drug resistance to bacteria previously not resistant to that drug. Misuse of the antibiotics can also result in the formation of antibiotic-resistant strains. Antibiotics must be administered at a high enough concentration and for a specific length of time in order to ensure the bacterial infection is completely eliminated. If some of the infecting bacteria remain, they will continue to reproduce and will pass on their drug-resistant traits to all future generations, resulting in the development of an increasingly resistant population of bacteria. Inadequate diagnosis of the actual infection can lead to the prescribing of an inappropriate antibiotic, further advancing the development of the resistant bacteria. Though the development of antibiotics has saved countless lives, now after 50 years of widespread use of a variety of antibiotics, resistance has become a challenge around the world. One resistant strain of bacteria is methicillin-resistant Staphylococcus aureus, more commonly known as MRSA. It derives its name from the antibiotic methicillin, previously capable of controlling an infection caused by this bacteria. MRSA was originally identified in 1961 and was first reported in the United States in 1968. Since then, it has become resistant to many other types of antibiotics. Vancomycin-resistant enterococci, known as VRE, is another type of common bacterial infection within hospitals, and it is of particular interest among researchers since it has the ability to transfer its antibiotic-resistant genes to other strains of bacteria like the previously mentioned MRSA. Resistance is also a concern within antibiotic use for livestock, which accounts for over 50% of the antibiotic usage in the United States. Some approaches to dealing with the problem of antibiotic resistance are to reduce improper uses by patients as well as incorrect diagnoses by doctors. Both of these accelerate the development of resistant forms of bacteria. The National Institute of Allergy and Infectious Disease, part of the Department of Health and Human Services within the National Institute of Health, has identified a number of high-priority areas of research, including in-depth study of the already present resistant strains of the organisms, improving diagnostic techniques, creating new therapies or treatments, including vaccines, and also improving the prevention and slowing the spread of the infectious agents.